अस्सलाम वालेकुम। इन स्लाइड्स में हम बात करेंगे सेंट्रल डॉगमा ऑफ मोलेकुलर बायोलॉजी वो क्या है एंड दिस इज एक्चुअली द गेट टू द gene expression. Central dogma of molecular biology, how we can define the central dogma of molecular biology, that is the flow of information, the flow of genetic information within a biological system. In this process, genetic information stored in the DNA is used to produce a functional protein. So there are different steps in this process. The key steps involve replication. Here you can see the DNA is replicated. That is also part of the central dogma. DNA makes a copy of itself to pass on the genetic information during cell division. And second important part of the central dogma is transcription, where this genetic information present on the DNA is transcribed in the form of RNA through the process of transcription. And in case of the third step, there is translation, where the messenger RNA molecule is translated into a protein. The messenger RNA molecule serves as a template for assembling amino acid into a polypeptide chain, which folds into a functional protein afterwards. So in 1958, Francis Crick originally described the flow of information as the central dogma. The DNA transfers its information to the RNA, which then directly controls the protein synthesis. The DNA also controls its own replication as well, so that is the central dogma. So in transcription, the transcription is a process of synthesizing messenger RNA molecule or RNA molecules from the DNA template using the rules of complementarity. Here you can see this is the rule of complementarity, which we already discussed in the structure of the DNA. A always make double bond with the T always make double bond with the A, G with triple bond with the C, and C triple bond with the G. This is called the rule of complementarity. The DNA information is rewritten, but it is same nucleotide language. RNA controls the synthesis of protein in case of translation, because information in the language of nucleotide is translated into information in the language of amino acid. So on the RNA there are codons, or genetic code is written on the RNA, which is anti-coded by the transfer RNA molecules which are charged with the amino acids to make a polypeptide chain in the ribosomes. The central dogma, the process of the central dogma is not one-way flow of information. Sometimes in case of some viruses like retroviruses, tumor viruses, and rose chromoviruses, the reverse transcription also takes place, and that is the function of reverse transcriptase enzymes that synthesizes a DNA strand complementary to the viral DNA. So there were two scientists, Tamin and Baltimore, who were awarded with the Nobel Prize for discovery of this reverse transcriptase enzyme and reverse transcription process. The central dogma of molecular biology, we'll talk about this process in detail. For example, RNA self-replication also takes place. Before we talk about DNA replication, we talk about transcription and translation, and now we can see that the RNA can self-replicate itself as well. The second modification to the original central dogma is the verification that RNA can act as a template for its own replication. So this process is known in small class of phages. These RNA phages such as R17, F2, MS2, and Q beta are the simplest phages known. MS2 phage contains about 3,500 nucleotides and codes for only three proteins: the core protein, the attachment protein, and a subunit of enzymes called RNA replicase. The RNA replicase subunit combines with the three of cell proteins to form RNA replicase, allowing the single-stranded RNA molecule of the phage to replicate itself. Similarly, RNA involvement in the translation. This process was discovered by the McCarthy and Holland in 1960s, which shows that under certain experimental conditions, denatured DNA, single-stranded DNA, could bind to the ribosomes and be translated into a protein by skipping this process of transcription. The experimental conditions usually involved the addition of antibiotics and that interacted with the DNA molecules or the ribosomes, and direct translation of the DNA, however, is not known in the cells in nature. So in next slides, we'll talk about ribonucleic acid, how it was identified, how it is different than the DNA, and what are RNA types. Thank you very much.